Okay. Good afternoon, everyone. Ruth and I are very excited to tell you a bit more about our ongoing project entitled Are We on the Same Wavelength? So on this image here, you can see two people who are interacting with each other. And while they are doing so, a process called biobehavioral synchrony is taking place. This biobehavioral synchrony framework refers to the temporal alignment of biological processes and behavioral patterns in interacting individuals. The biological part of the definition refers to the temporal alignment or synchronization of, for example, heart rate, brain activity, and even hormone levels. Whereas the behavioral part refers to the coordination of behaviors, movements, actions, such as smiling at each other, engaging in eye contact, and imitating gestures. As of birth, children and their parents engage in coordinating their behaviors and biological processes. And these early forms of biobehavioral synchrony really form the foundation for normative social development. So synchrony plays an important role in social bonding and emotion regulation. And a key player in all these processes is oxytocin, also referred to as the love hormone. So biobehavioral synchrony, especially during mother-infant bonding, also shapes all later attachments in life, including romantic lovers, friends, and even strangers. And this is why biobehavioral synchrony is also evident across these other attachment constellations. Now let's have a look at autism. So autism spectrum disorder is a neurodevelopmental condition that is characterized by difficulties in social interaction and communication, as well as the presence of repetitive or restrictive patterns of behaviors, interests, or activities. In terms of social interaction difficulties, we know that these individuals may struggle with back and forth conversations or with engaging in eye contact. However, in terms of biobehavioral synchrony, not much research has been done yet, but the few studies that have looked into this generally report reduced or atypical synchrony in autism in terms of motor, conversational, physiological, and neural synchrony. As I already mentioned, oxytocin is a key player in regulating biobehavioral synchrony processes, but also social interactions in general. The neuropeptide oxytocin is produced in the hypothalamus and exerts peripheral as well as various central effects. Among others, oxytocin facilitates social bonding and attachment, for instance, by altering social decision-making, but also modulating the neural processing of social stimuli. Among others, and according to the social salience theory, oxytocin can increase the salience of socially relevant cues. So this means that oxytocin can orient the attention of the observer towards this particular cue. So if the oxytocinergic system is involved in facilitating successful social interaction by mediating biobehavioral synchrony, and this synchrony in turn has been demonstrated to be atypical or reduced in autism, the question raises whether we could therapeutically target the oxytocinergic system in order to promote biobehavioral synchrony and therefore social interaction as well. In theory, we can do this, even in various ways. So firstly, there's a large body of research looking into the exogenous administration of oxytocin. We know that oxytocin administered via a nasal spray can increase endogenous oxytocin levels. So it means that it can cross the blood-brain barrier. Additionally, single-dose administration studies in autism show that oxytocin can enhance social cognition. However, more recent studies also point to the importance of the context in which oxytocin is administered. So to promote social abilities in a person, researchers actually suggest to combine the oxytocin administration with a social context. So in this regard, we will combine oxytocin with a mirror game in our study. So during this mirror game, participants imitate each other's body movements, which is not only fun in nature, but also highly social. And additionally, these synchronous body movements on their own already increase endogenous oxytocin levels. So against this background, we have set up the OxySync study, and Ruth will now tell you a bit more about the study design.
So, the OxySync study consists of 2 visits at our lab. During the first visit, we are screening the children based on specific inclusion and exclusion criteria. This involves questionnaires and an IQ testing to determine if they qualify for participation. We are including children, both boys and girls, aged 8 to 12 with an average intelligence, so an IQ above 70. Our goal is to recruit 140 children in total, including 100 children with a diagnosis of autism and 40 neurotypically developing children. Each participant will complete a pre- and post-measurements in which we are collecting biobehavioral data from both the child and the experimenter. We are also collecting saliva samples at the start, the end, and in the middle of the session to measure some endogenous oxytocin levels. In between these pre- and post-measurements, the children with autism will be randomly assigned to one of the intervention groups, while the neurotypically developing children will take a break. The children with autism will either receive a nasal spray with oxytocin, the cuddle hormone Laura already explained something about, or a placebo one. This nasal spray is then paired with a mirror game. This mirror game can be either social. This is where the child is interacting with one of our researchers, and another one is a non-social one. This is a game where they are playing together with the screen. To measure this biobehavioral synchrony, we are using an innovative two-way real-life video feed setup. This setup is designed by Professor Roy Hessels from the University of Utrecht and allows for a natural conversation between the child and the researcher, in which the researcher will interact as a stranger. They will engage in different interaction paradigms. We will start with the baseline measurement. This is a resting stage in which they are both focusing on a black cross. Then they are instructed to look at each other without speaking. This is then followed by an instructed looking paradigm, in which both the child and the researcher are instructed to look at their mouth, each other's mouth, nose, or eyes. And then they are engaging in a natural conversation about familiar topics, like the school context, home, or hobbies. During these interactions, we are simultaneously measuring the behavioral and biological measurements in both the child and the researcher. Behavioral measures include eye tracking, this to study gaze behavior. On the biological side, we are including some neurophysiological measures, and these include the electrocardiography to monitor heart rate, the galvanic skin response to assess skin conductance, and the electroencephalography to capture the neural responses. In this randomized, placebo-controlled, partially blinded clinical trial, we aim to address two large objectives. The first one is that we will try to quantify the biobehavioral synchrony between a researcher, who will be a stranger, and the children. These children will be the ones with and without autism. And then we are also looking at an intervention effect. This to explore the unique and combined effect of a single-dose intranasal oxytocin administration and the dyadic mirror game, specifically in the children with autism. This with a larger goal to help children with autism connect more effectively with others and to make them feel more comfortable during social interactions. So, what do we think that will happen? We hypothesize a reduced responsivity and synchronization towards the biobehavioral signals of the researcher in autism compared to typically developing peers. When we look at the intervention effect, we think or we expect enhanced responsivity and synchronization in autism following the oxytocin administration, the social mirror game, or a combination of both. So this was the OxySync study. I hope you found it interesting. Thank you for listening.
Questions for Ruth and Laura? Anyone? I do have a question. So why were you controlling for the IQ for the children? Like, why is it relevant to this specific measure?
So the question is why control for the eye track?
Yeah, because no, the IQ.
For the IQ?
The IQ score was one part of the inclusion criteria, so...
Yeah, we think it's indeed important to look at the IQ, to take it in as like some behavioral measure that is different in all the children, because you have high-functioning children with autism with a very high IQ, but also a bit lower, and we think that this can be have an effect on maybe their functioning and their skills to like compensate in social context. I don't know if you want to add something.
Okay. Yeah, I was thinking because there are other things I would have expected to interfere, like a body mass index, things like this, or literacy of the child, but I don't know.
I also maybe want to add that in these types of studies, it's often done to match both groups on
Okay,
IQ.
okay. Thank
Just,
you.
yeah.
Sorry, Harish.
It's super interesting. With the oxytocin, like when you kind of get the spray, do you actually feel any changes, or is it kind of like, do you actually get kind of some kind of a high? Like not like high, but do you feel any kind of changes in your own state?
So the oxytocin or the nasal spray is only administered to the child, not to the researcher.
Yeah, I understand.
But yeah, could be a new future study. But we have had some children who stated that they were like more happy, or we asked their mothers, and they were like, they're more. It seems like their kids were more happy or more enthusiastic.
The reason why I'm asking is I kind of wonder what's the good placebo. It's almost like with the placebo, it's kind of, because I wonder like what's really about the oxytocin rather than about feeling happy and kind of, what I'm trying to
It's also a bit at this stage difficult to disentangle when we ask the parents, what do you think? Is your child looking differently now, behaving differently? Also because it's a single dose, and secondly, either way they got a nasal spray. So and they took part in a clinical trial. So yeah, it's difficult to disentangle. Yeah.
Yeah, but that's super cool. Thank you.